Good morning to all. So yesterday we started, it was about so we completed successfully, that is about the transcription, translation and genetic code. So genetic code we completed in a previous class. So that was the information which was coded from the genes, that is the DNA. So that was mainly in the form of codons inside mainly in mRNA. So the mRNA codons are going to translated in to the tRNA. The tRNA may contain the anticodons for those codons. So the codons which are presented on the surface of the mRNA are so going to be bonded with the anticodons of so another one, that is a tRNA. So that was mainly genetic code. That was the genetic code was commonless, and the genetic code was so universal, and that was a degenerative state, and so that was so polarity that may also so that was the strand was synthesized in 5' to 3' end direction. That's why that was also considered as the polarity. So those are the some of the properties of so these mainly genetic code. So we are completed in our yesterday class. Today we are going to discuss about the another topic, that was the lac operon concept. So it was by the Jacob and Monod. It was by the two scientists, that was the Jacob and Monod, and the lac operon. So the lac operon is a set of genes. Operon is a set of genes or DNA, a set of gene and a part of DNA. Operon is, so that was a set of genes and also considered a part of DNA. So that's why the operon, so these was mainly under the, so by the presence of the lactose. So that is lactose is one of the lactose. So lac means so it was lactose. Lactose was one of the disaccharide. So lactose one of the disaccharide. It was furtherly divided into glucose and galactose. So mainly at the time of occurring of the process, that is a replication of DNA, transcription level and the translation level, what happens is so some of the changes are occurred. The wrongly coded amino acids are also going to be attached with the tRNA, and that was takes place at the time of the replication process. So in the replication process, the DNA was going to be, that was so that was going to be divided and either two forks, so in a Y-shaped like structure and a double helical structure. So that was going to be again coded with the nucleotides, that was bonded with the amino acids. So nucleotides, at the binding of nucleotides in the replication level, the nucleotides are wrongly coded. So then what happens is that was mainly observed and that was removed from the particular DNA structure, and so the new nucleotides and so the correct sequence was added to that, and that problematics are also occurred at the time of replication level and the transcription level. The transcription level, what happens is so from the DNA to mRNA, the information got transmitted. So that is a transcription process. At that time also some of the problems are occurred. So otherwise the translation process means at the time of mRNA to tRNA, the information, that is the codons, are going to bonded with the anticodons. So any wrongly coded amino acids are bonded with the anticodons, then that was completely formed a that was a false amino acid. So that should be problematic. So that's why, to avoid that problem, so any mainly at the time of replication level, at the time of transcription level, at the time of translation level, so the gene regulation was takes place. So that was a gene expression. The gene regulation, and it is completely called as gene expression. So at this time what happens is so any problems are occurred, any so the wrongly coded amino acids or any, so these problems are mainly associated with the So these at the time of replication, transcription and the translation, so those are process what happens is leads to cause the so the gene regulation that should be needed, the gene regulation process. So that's why the lag operon concept is also called as gene regulation process and the gene expression. So it is we are going to discuss about the lag operon concept. The lactose mainly inside the transcription process inside the prokaryotes. So these lag operon concept are mainly applicable to that and also to the eukaryotes. So the eukaryotes, that is the transcription that was only so initiated inside the prokaryotes. So the only single mRNA is presented at there. So the multiples are presented inside the eukaryotes here. So at the time of prokaryotes, so what happens is so mainly the E. coli bacteria, the bacterial cells are cultured in a media, mainly the lactose media. Then what happens is so those are going to digest, so those are easily going to absorb the lactose and converted in the lactose into the glucose and the galactose. The lactose digestion, the mainly inside the prokaryotes. So that was applicable in the presence of the lactose, the operon concept at the time of so these are levels. So then what happens is the gene regulation should be needed, that is completely considered as a lag operon concept. See this, see the variation between these two. So mainly these are mainly contain the so the lac operon concept by the Jock and Monod by the two scientists. So it may be mainly present. So that was in at the time of in a prokaryotes transcription. So in prokaryotes transcription, that was the bacteria are presented. The prokaryotes, the main example, the bacteria. So the viruses are prokaryotes, and so we are discussing about the E. coli bacteria, the lac operon model in the E. coli bacteria. So the E. coli bacteria for the mainly depends upon the cultured, we cultured the bacteria in a particular media, then so their number was greatly increased in the presence of lactose. So in the they introduce the lactose into that media, cultured media, then what happens is the growth rate of E. coli bacteria was greatly increased. So due to the lactose utilization, the lactose was divided into the digested into the that was converted into the that was glucose and the galactose, and that was a growth in so the number was that was greatly increased. And here, so see here the lac operon concept by the Jacques Onard, so that was mainly depends upon the promoter region. So it was so the I, so it was I was considered as a regulatory gene. So it was considered as a regulatory gene. And it is a promoter region and it is a operator region, and so these the promoter and the operator are mainly helpful for the continuation process of the transcription. At the time of the initiation of the transcription, the promoter and the operators or the genes, those are participated for the so the initiation also for the transcription process. And so ZYA, so these are considered, see here the lac Z and the lac Y and the lac A. So Z was considered as a beta galactosidase enzyme. So these are the structural genes and also considered as the enzymes. So these are the Z, Y, A are considered as a structural genes and also considered these are the enzymes. So that is Z was a beta galactosidase enzyme. I was a regulatory gene. So it code for that was a replication process. It was a code for the replication process, that is so that was a regulatory gene, that is I inhibitor, is also called as a inhibitor, and operator region, promoter region, and so beta galactosidase. Beta galactosidase is the enzyme which was going to hydrolyze the that was a lactose into the glucose and galactose, glucose and galactose. So that was a beta galactosidase is one of the structural enzyme, that is a Z and also considered as a lac Z here. So this lac Z was a beta galactosidase. It was an enzyme mainly used to hydrolyze, means going to digest, hydrolyze. So it was going to hydrolyze and converts that was a lactose into the glucose and a galactose. That was the main functioning of these galactosidase. And the lac Y was considered as a permease enzyme. So these permease was so it was give the permission, the permeability to enter the beta galactosidase into the cell. It was going to allow these beta galactosidase into the cell. Permease is another enzyme. It may allows these beta galactosidase into the cell, means it may give the permeability, acceptancy to enter the beta galactosidase into the cell, these enzyme into the cell. And A, so it was a transacetylase. So it codes for, so that was a translation process. It was a mainly, so it was a codes for the transacetylase translation process. So the acetyl group, it was going to transfer acetylase. So acetyl group was going to be translated. That's why it is a transacetylase. The acetyl group was going to be translated. So the information was coded for this. For the after the completion of the transcription, the translation process was started. It was also going to code for the translation information, and it was also used to translate the acetyl group. So these three are the enzymes. These are the lac Z, lac Y and lac A. Beta galactosidase, it was going to penetrate means hydrolyze and converts the lactose into the glucose and galactose, that is simple form. Why? Because the lactose is the disaccharide that was going to convert into the simple form, that is the monosaccharides or the glucose and the galactose, and the permease is the enzyme. So it was maybe allows the beta galactosidase into the cell, and trans-stylase, it was going to translate the acyl group and also coded for the information of the translation. And the remaining, see here, the inducer, see here, in absence of an inducer here. So here there is no inducer and mRNA, and a repressor protein is presented here. So repressor is a protein, and see here the inducer was going to bond with the operator region. The inducer was going to, actually what happens is so the transcription was going to be inhibited by the effect of repressor protein. The transcription process was inhibited, stopped. So this process was mainly occurred. So any the wrong sequences are added to the transcription process, any problems are associated with the transcription, then only then what happens is the temporarily the transcription process was going to be inhibited. So at that time what happens is by that reason then immediately the process, the inhibition process was occurred. So the absence of inducer is a protein, repressor is another protein. So these repressor was going to bond with the operator region. So then what happens is so if there is no inducer, the repressor was going to bond with the operator region. If the inducer is so released here, the inducer was came here, then immediately it was going to bonded with the repressor protein. The inducer was going to bonded with the, see here, inducer bonded with the repressor protein. Then what happens is the root clearance. The root clearance the mRNA, so that was going to be RNA polymerase was going to bonded here, and then the what happens is the transcription was again continued. Temporarily the transcription process was stopped. That was mainly occurred with the help of, so these are an absence of inducer. There is no inducer for the occurring of so any the temporarily the transcription was stopped here. So by that reason what happens is the mRNA is RNA polymerase can plays a major role in transcription process from the DNA to mRNA, the information coded. So at that time the transcription was temporarily inhibited. So by the, so mainly for the repressor protein, the repressor was going to bond with the operator region, then immediately the process was completely transcription was stopped. So by that reason the inducer was released, inducer was reached here, and it was going to bond with the repressor protein, then only the repressor was removed from the operator, then the transcription was continued here. See here, the RNA polymerase, it is a RNA polymerase enzyme, it is a DNA that at the time of transcription it is a, so it was a mainly, so you see here it is a mRNA here, so it is a mRNA here. So promoter region, operator region, lag Z, lag Y and lag A. So these are the structural genes. The transcription was inhibited here. So it is a lag repressor. So see there, that is a repressor protein. So the lactose is presented here, and operator region. See here, the operator region was temporarily that was inhibited. Why? Because this repressor was going to bond with the operator region. See this. So it is operator region. So the lag repressor was going to be bounded here. Then the what happens is the transcription process was temporarily inhibited. So then what happens is so the immediately in the presence of lactose, so by the presence of lactose, then immediately the lag repressor is another protein. So see here, there we considered, so then we so explain about the repressor, that is a simple repressor, but that is a lag repressor in the presence of the lactose. So that was going to be bonded with the, so these the operator region that was going to be bonded with the repressor. The lag repressor is that was a inhibitor here. So it was an inhibitor for the transcription process. The temporarily the transcription was stopped, inhibited by the effect of the repressor was the lag repressor was going to be bonded so with the operator region. Then what happens is the transcription process was stopped, inhibited. So then immediately, so in the presence of the lactose and the lag repressor, so it was so the repressor was reached here, that was going to be bonded with the repressor bonded with the, so that was a lag repressor, so with the lactose. So then immediately what happens is so it is a RNA polymerase is presented here. See this, The ball-like structure is the RNA polymerase. So, these RNA polymerase, when the operator region was cleared, so the root cleared, the operator region was then becomes there. So, those also the operator region was bonded with the repressor protein. Here also the operator was bonded with the repressor, that is a lac repressor. Then immediately the transcription was stopped, inhibited. So, the root clearance that was mainly by the lac repressor was inactivated by that was bonded with the lactose. So, the lactose was going to bond these repressor protein, that is a inducer. The lactose was act as an inducer. So, it was as an inducer. So, it was when it was going to bond with the lac repressor protein, then immediately, so it was removed. So, the lac repressor was going to bond, that is the lactose was bonded with the. So, see there, the inducer was bonded with the repressor, then the operator region was become cleared. That was helpful for, that was bonded with the RNA polymerase and the transcription continued. Here also the process is same process. The process was nothing but here also, that is the lactose was going to be reached here, released here, and the operator region was already bonded with the lac repressor. It is an inhibitor. The lactose was a inducer. See there, when the inducer was reached, those inducer was going to bonded with the lac, that was a repressor protein, lac repressor. Then the lactose and the repressor are bonded with each other. Then the operator region, from the operator region, the lactose was going to remove the repressor. The lac repressor was going to be removed. So, by that reason the operator become cleared. The operator region was then only the operator region was bounded with the RNA polymerase enzyme. Now, the RNA polymerase enzyme, so it was going to bond with the operator region. So, then the DNA to mRNA, the information got transcripted. So, this process was continued as that is transcription activated and the transcription continued. So, these process was mainly considered here as that was a lac operon concept. So, it is today's topic, it is completed. So, we will meet tomorrow. Thank you.